Hello. Thanks everyone for coming. I'm really excited to talk to you today about the work I've been helping do to scale phage therapy, starting from Phage Directory about 6 years ago, all the way to now where I'm working with Phage Australia. So 6 years ago, my friend Jan and I noticed a problem. So people were using Twitter to find phages for their patients. Doctors were kind of out of options, and this was starting to happen even in the USA where I was. And so there's me on the left in the lab. I'm a grad student at this point, 2017, just doing phage work, not thinking about phage therapy. And then Jan, I met him through swing dancing, and he's there at the bottom, surrounded by computers, as is his way. And so he kind of saw this tweet with me and had the idea to create a system where we could help people find phages for their patients, so they don't have to just hope that the right lab sees the tweet. And this tweet was all around trying to help Mallory Smith, who's here on the right. And unfortunately, she passed away before they could actually get phage therapy for her. So this really is what spurred on Phage Directory. And so what it's become is a system that from then and still is today, kind of works the same way. So we have a phage alert system where a physician who has a patient who's tried all their other options, antibiotics have failed, they can send out a phage alert to our network of researchers, and they're all around the world. There's about 300 labs that regularly get our messages, respond, and when they have capacity to help with phage screening and phage purification, whatever is needed. And so we've done about 50 of these phage alerts to date that have actually gone all the way through, and 4 of them have led to successful treatments. So 3 humans, 1 sea turtle. The sea turtle is a story for another day, but the system works just the same if you're a vet. And so one of those humans who was treated, I'm going to get to next, is kind of what brought us to Phage Australia, or to move to Australia. So one of the patients that we actually ended up helping came from a request from the Iredell lab in Sydney. So Jan and I were living in Georgia at the time. We had never had one of our alerts come from Australia, but thought, well, doesn't matter where they're coming from. Let's go ahead and see if anybody will volunteer to help. And so fortunately, we had a huge response to this one, and there was 12 phage labs around the world. They all came from different countries, so 12 countries, offering to receive the isolate and test their phages. And so the team on the ground, so John Iredell, pictured here, Ruby Lynn, and Amine Katami, they were able to make everything work on their end to actually treat Danby, who's 7 years old at the time, and treat her in about 6 weeks from the initial phage alert. They got their phages through the community, and yeah, they were able to save her leg from amputation. So this is a really cool success. And for us on Phage Directory side, we had never had it where it actually all the way went through and really treated a patient. So we were really excited. And then the team in Australia was able to bring in about $3 million in grant funding off the back of this success and created this consortium called Phage Australia. And they brought us in as a partner so that we could help them scale this to more patients. And so when we got to Australia, this was 2022, early last year at this point, we kind of came into this really unique opportunity from a data collection point of view, because the team had just gotten this clinical trial approved called STAMP. It's a standardized treatment and monitoring protocol for phage therapy. And so it's unique in that it allows every patient in the trial to be treated with bespoke phages, but the standardization is around the dosing and the administration and the data collection. So this is really interesting, and this is kind of what we walked into. They had just gotten it approved, had received national ethics approval, endorsed by ID physicians across Australia. Everybody was kind of ready to start treating patients under this framework. So that's where we were. And even though I had spent 4 years out of the lab working on Phage Directory, I kind of jumped back in because, yeah, that's what was needed. There was right away patients kind of lined up, as everybody here is probably familiar with. there's no shortage of patients that are waiting for a phage therapy. And so I was kind of one of the people in the lab that was set out to make the first batch. And so this first case was nice because it was one patient that had an E. coli infection, and we had a bunch of E. coli phages. Because we only had to focus on one case at a time at this point, it was straightforward. We followed basic lab techniques from the literature. We used the phage on tap method by Jeremy Barr's team that have been used in the Patterson case. We used small scale batches. We got rid of endotoxin, and we sequenced it, and we felt good about this first batch. And it actually did lead to a successful outcome clinically, microbiologically, which was icing on the cake. But we also kind of felt good about having like our first version of the process of making phages for these patients done. At this point, though, the floodgates started cracking open. So doctors from around the community and also around Australia started kind of picking up the phone and calling John and saying, well, I have a patient who could benefit from phage therapy. And how soon could you make phages? And so we're not some phage making operation. We've done it once, but, we picked up the phone and had these calls. And it wasn't long before the system kind of started to strain. This is the kind of phone calls we would have. It made me really realize that, the complexity of all the different interactions when you have a phage treatment you're trying to coordinate it just gets rapidly scales up pretty quickly. And so, things like we have too much endotoxin produced, like it's not looking like the last time. I don't think we can make phages as quick as last time. Or, doctors calling us and saying like, did you receive the isolate from my patient? But at the time, we didn't really have a system for tracking which isolates were coming in according to which patient. So it's a lot of conversation like, oh, was that the case that you sent last week, or was this a different Pseudomonas case? Things like that. Just your basic workflow and communication tangled web. But, we try to roll with it. And so eventually we were able to kind of, because we had, clearly we're not going to be able to have the luxury of doing one at a time anymore. We got to create a system that is going to help us internally manage which phages we're giving to whom, and are these phage preps each safe? And, the worst thing we could do is go too fast and try to treat too many patients at once, mix up which phage is for which patient, mix up which batch has already been purified, which hasn't. So we definitely felt strongly about kind of ironing out what is our process and how are we going to deal with this. And so this is kind of just a quick bird's eye overview of our current process. It looks a lot like probably most other phage therapy centers are using these days. So there's a pre-matching stage where this is phage isolation, phage characterization to some extent, identifying the patient, getting them clinically kind of approved for phage therapy with our clinical team, and then receiving their isolate for testing. Then we have a matching phase where we check of our phages that we have and those of collaborators, do we have a hit or multiple hits? Doing some kinetics assays and antibiotic synergy if we can. And then selecting phage candidates, getting them purified, which would be the next stage. Removing endotoxin is our crucial metric. And then creating some kind of documentation of the quality of that batch. So sequencing the batch, performing endotoxin testing, things like that. And then the last stage would be the treatment and monitoring. So this is where we are sending the clinician, whoever's the treating physician, the phage itself, the vials of phage, and some kind of description of what they're getting and why it was chosen. And then giving them the patient, or the patient gets the phage, and then there's a monitoring stage, this last piece, which is what the STAMP trial is really all about. And I won't really get into it today in detail, but my colleague Alex will be talking about this later in the conference. But yeah, that's when we're collecting blood samples, tracking phages and strains over time during therapy. And so, yeah, we really, pieced this together over the last year. And some of the things that we found are crucial are things you wouldn't think of. Like when you think of phage therapy challenges, you think of like, well, the phage and the patient, but there's a lot of things that you wouldn't think of, like standard protocols and reporting. Like if we're going to have multiple people doing this in a team environment, knowing that both of the postdocs in the lab who might be doing one of these are using the same titration protocol, and are we reporting the same things every time? So we've been coming up with some standard protocols and standard worksheets that go along with each protocol. And then what thresholds matter, like along with piecing together the steps, we need to know at which step, like when do we go or not go to the next step? And that has been a lot of decision making with our team and looking at the literature. And most of this, it's all out there for the taking, and we are just pretty much stringing together what's been published to date and just making decisions on what we're going to use, what we're not. So high enough phage that we have enough after the purification process, low enough endotoxin that it's safe enough to meet FDA guidelines. And then we're also, we found that a single source of truth of where we're actually writing everything down is crucial, because we're getting phone calls all the time, and any member of the team could get one of these and needs to be able to find the information, even if they're not the person who just did that titer. They need to be able to know, is that phage on track for that patient? And we've also found it's been helpful to divide up roles. So having one person responsible for diagnostics and one person responsible for production and purification, and defining the handoff points between when we have a phage that's ready to move on to production. What does the production person get from the person who checked did the diagnostics? So that's down here, this QR code. If you want to learn more about our process, I've been blogging about it at phageaustralia.org/blog, and just kind of blogging about all kinds of sides of this system and how we've been piecing it together. So around this point, this is about last fall, if you're not in Australia, around September, October, we started hearing that from the regulator, the health regulator, the TGA in Australia, and also hospital drug committees who are receiving our phage preparations. People basically want to know what is in our phage preparations, and also the regulator actually was asking us, what do you think about how we should regulate phage products? And that's a conversation that we've been seeing as a big opportunity that we get to participate in. Similar, I think, to what's happened in Belgium and what's happening in the UK lately, where regulators are actually reaching out to phage labs and clinicians and producers to have the conversation together about what makes sense in our specific local context. And so we're hearing these questions, and now it's suddenly like, okay, we just got our phage making process straight, and we're making our phages for patients, but we also have to focus on how do we document them and how do we make them formalized in a way that's going to meet what these groups need to know. And so this led us to create a set of 4 documents. We kind of call them the 4 reports colloquially. And we came up with these by looking at who all needs to know what and at what time during the whole process. And so the first is the phage recommendation report. So this is something that the physician gets at the bedside. They see it like, it looks like an antibiotic susceptibility report that they would be used to. We developed that with our main clinician PI of the Phage Australia project, John Iredell. And so we're able to kind of make a report that is something that the physician is going to be familiar with and exactly what they need to know about the phage they're about to administer. Like, why was this phage chosen? Then we have 2 safety sheets for genomic safety sheets, essentially. So the phage safety data sheet, we call it, is for looking at the phage genome, things that, you know, the field has established for quite some time now. Things like, are there lysogeny genes? Are there AMR genes? Are there toxins? Anything that's a red flag genetically that would make us not want to put this phage in a patient. We do that up for every phage that we make. Then there's a bacterial safety data sheet that this is for the propagating strain, so the one that we use in production. We look for red flag genes from that genome as well, such as prophages or AMR genes, or just nasty virulence genes that we don't want to see going into the patient. And so we also give that every time. And then the batch safety data sheet, this one's really directed at the hospital drug committees, and also the TGA wants to see more here, where it's, of course, the final batch. Yes, it has this phage in it, and it was made on this bacterial strain for this patient, but physically, what is the composition of the batch? Does it have, like, what amount of endotoxin does it still have? What's the pH? And have you done sterility testing to prove that there's no contaminants in there? And of note, all of this, again, I hope this is a theme that you can see. We did not come up with this ourselves. We took from what's already out there. And luckily, there's been groups like Sciensano and Queen Astrid Military Hospital in Belgium, who you would have just heard Jean-Paul talk about. They were our inspiration for the batch safety data sheet, and we pretty much just followed what they do for issuing what they call a phage passport. And then Taylor Labs in Texas was the inspiration for this first report. They have a physician desk reference that they give to their physicians before they hand over the phage. So it's all been kind of just us trying to be practical, give the minimal information that is needed by these different parties, and also, crucially, not be overburdening on us because we have to once we say that we're going to do these reports every time, that means we have to do them every time. And so we put forth this package, and it ended up being, it's about 76-page PDF for every patient, and put this forth to the hospital drug committees at the Westmead Hospital and other local ones, and kind of crossed our fingers that it would be acceptable. And luckily, it seems to have gone ahead. The drug committees are happy with the amount of information we're giving. So this is going to be functioning as our standard set of documents, and that we kind of know exactly what we're reporting every time we make a phage. So yay, success. But now we're back to the lab. Now it's time to treat more patients. So to give you a sense of kind of how fast we're able to go, over the last 8 months, we've made phages for 7 patients, and they've all been treated in that time. So it's just under 1 phage a month. But what we can't do is 10 phages a month. We can't treat 10 patients a month. We have a fixed head count, a fixed budget at this point. So how can we possibly, should we keep going at our current rate and turning away all the people on our waiting list and all the phone calls we're getting from doctors, or can we try to look at making this process more efficient? So looking at our process, we're seeing about 150 hours of work per patient. Some of that's lab work, some of it's not. So where is our time going? What we identified, even though most of that time is lab work and there's a lot to be done automating lab work, there's also about 20 hours of paperwork per patient. And so we figured we can kind of focus on this as one way to make things more streamlined without having to buy new equipment or robots. And so we're using kind of a different kind of robot, which is something you might be familiar with now, ChatGPT. So we have all these new large language models that are available for people to use and play with and connect with their own systems. And if any of you have been playing with it, you'll know what I mean. And so we're starting to think a lot about how we can use these tools to actually apply them to our problems here and expedite the paperwork steps so we can treat our 10 patients goal per month without 200 hours of paperwork. And yes, the way I think about it is all this data on the left that comes from all this messy lab work and sequencing, purification, plaque assays. How do we turn this into these 4 reports without, hundreds of hours of work every month? And as a toy example that we have been putting together to kind of get going on this is a label maker that makes our phage batch labels. They have specific defined things that need to be on them. And this is like a free text box that using ChatGPT in the back end we can actually turn this text into the label just in one click now. So it's able to extract the insights that are needed based on what it needs that we tell it. And then we have a label in a few clicks. And so I hope that this illustrates kind of, and maybe others here already feel and already see the potential, but I want like all of our free text to be going into the actual reports next. And so that's been really exciting. And we are putting our tools on labspace.ai. So just a little sandbox playground website where we're just going to be adding all of our AI tools. You can use this QR code to try the label maker. But yeah, there's going to be more to come, maybe even when this, I'm recording this 1 week before you'll be seeing it. So who knows what'll be on labspace.ai by next week. So it's been really exciting. So kind of to come to a close, really everything, it's been a whirlwind year and a whirlwind 6 years, really, all around trying to make phage therapy available for patients. Started with Mallory, and there's just so many more like her. It's all about being fast enough that we can maximize the patients treated, but being slow and careful and cautious enough that we don't screw it up for everybody and cause something that happens as bad to a patient. So we really can't move faster than our systems will allow. We need to keep patients safe and continue earning the trust from regulators and from clinicians. But can we see if we can maintain that confidence, but keep moving and keep trying to increase the amount of patients we can treat? So better processes and better tools are going to help us do that, and focusing on documentation without overwhelming ourselves and making it impossible to move forward. That's the balance that we're dealing with. So thank you so much, everyone, for listening. Thanks to my co-founder of Phage Directory, Jan. He's here. Yeah, everything has been, I never would have started this without him. I would have just kept doing my plaque assays in the lab. So I'm always grateful to have had my mind open that you can actually build these kind of systems and you don't have to wait for somebody's permission. People are actually, they love it when you bring them something that's going to help. And Phage Directory is a volunteer, like we have a ton of volunteers that help us with everything from social media to putting together a newsletter every week, to coming to conferences like this one, where two of these, Atif and Sheetal, are in attendance. If you can find Sheetal, she'll be there in person. So that, yeah, they help us report on conferences and keep the Phage community up to date. So we love our volunteers, and it's just so cool that you guys keep helping us. And the Iredell Lab, this is kind of the team that we've joined here at Phage Australia. They've spearheaded that. Awesome people. It's been so cool getting so much closer to the actual nitty gritty of it all this last year and feeling what it's like actually on the ground, because I had it easy when I was just email connecting people and letting them share phages and just hands clean. Hopefully you share the right phages and everybody's happy, but being in the lab and being on the side of receiving all these phages, it's been completely eye-opening. And John, Ruby, and the team have been so awesome to bring us into the fold. Importantly, there's a million QR codes here for you to use your phone and grab these websites. So I blog about making phages for phage therapy, about quality checking, about how we're stringing together our protocols on the Phage Australia blog and on our Capsid and Tail blog, for which is the Phage Directory's weekly phage newsletter. Jan also blogs a lot on that about phage data and database systems, how he's building collection systems to collect clinical data, which I didn't talk about today, integrating that with the phage lab data. So if you want to read about that and get kind of an intro primer to how a data person and a software engineer thinks about how to build these systems, here's your QR code for the Capsid and Tail newsletter right here. That's the one. Then I said that labspace.ai is our little AI tool playground. You can recommend tools and we'll try to make them. And you can come try the label maker and other ones we have. We're also making like grant writing tools and kind of toying with things like that. If you want to list your lab on Phage Directory, here's the QR code for you. We have about 500 phage researchers listed, and you can also sign up to hear about alerts so your lab can help with the patient cases if you want. And lastly, next 2 months from now is the Evergreen Phage meeting. And so I am helping organize. Jan is doing a huge amount of the work behind the scenes making this conference possible. So we hope to see you all there. If you want to hang out, we're actually finally going to go fly to a meeting in person, which hasn't been possible since we moved to Australia. So really excited to be able to be there. And I wish I could be there at this one where you're all at in Paris, but it's so far away. So thanks so much for listening. Happy to take any questions.